First of all, we have to disinfect the bench and our hands. So using a disinfectant solution, just wash the hands and disinfect the bench. This removes any contaminating microorganisms that may be present. In this experiment, we're going to use broad bean seeds. Broad bean seeds, when they come out of the packet, are dry. They're extremely dry. So to allow the enzyme that's in these seeds to work, they have to be soaked overnight for about 24 hours, 48 hours in water. These seeds that I have here have been soaked overnight in water. So they've absorbed water, and now the enzymes are ready to act. The enzyme we're going to investigate is amylase. These seeds have the same enzyme that we produce in our salivary glands. And amylase breaks down starch, so it digests starch. The enzyme is amylase. The starch will be present in the starch agar plates. Here we've got agar with starch built into it. So the enzyme amylase, hopefully, will break down starch in the agar plates. As a control, we take one of the live seeds and we place it into boiling water. And we let it boil for about 10 minutes. Boiling it will denature the enzymes. The seed will be dead, and that will act as a control with the denatured enzyme. After 10 minutes, we remove the heat. So we'll pour away the boiling water. And we'll take the seed, and we'll put the seed into disinfectant. We'll also put a live and unboiled seed into disinfectant. So the two seeds, the control seed, the boiled seed, and the live or unboiled seed are left in disinfectant for about 10 minutes to kill any microorganisms on the surface. After soaking the two seeds in the disinfectant for 10 minutes, we want to sterilize the forceps. So we turn the bunsen to the hotter flame, and we just sterilize the forceps. If I just drop this into water just to cool it for a second. Now, if we take the dead seed, which is sterile, and the forceps, which is now sterile, and we take out the seed, and what we want to do is split the seed down along its length. Using a backed blade for safety, and it's a new blade, so it's sterile in itself, we want to split the seed down the middle. Splitting the seed will allow the enzymes to come in contact with the substrate in the dish in a moment. So we split the seed, and this is the boiled seed. So we want to put the split seed face down with the cut surface facing down so that the enzyme can come in contact with the starch. So we just place it face down onto the agar, and the same with the second piece of seed. Place it face down on the agar like so. We'll leave that for 2 days in a warm place. We now want to do basically the same with the unboiled seed. So again, I'll re-sterilize, just to be sure, I'll re-sterilize the forceps. And again, if I just cool it quickly, take out the live seed, and repeat the process. Using the sterile backed blade, just slice down through the seed to expose the enzymes. Again, we'll place this face down so that the amylase in the seed can come in contact with the starch that's in the agar to allow any reaction to occur. As we open the lid of the Petri dish, we open it for the least possible time and the least possible distance to prevent contamination from other microorganisms. Again, we leave this for 2 days in a warm place, room temperature, about 20 degrees centigrade. We now take out the live seeds and dispose of them. And again, we flood the dish with iodine solution. Having added the iodine, we swirl it around, and then we see the result as follows. Around the outside, blue-black, showing starch is present. Underneath the live seeds, there are clear patches showing that the enzyme amylase has broken down the starch. No starch present, indicated by the 2 clear patches.